Hello, my name is Kenan Menguc. I will presentation our research. Our research is the subject. Our title is the name Decoding the AI Arsenal of Bioinformatics Hierarchical Clustering of 153 Tools for Smarter Selection. Motivation, uh, rapid growth of AI tools, uh, difficult tool selection, need for quantitative classification, not only for normal tools increase number increase our life, also health tools also increase, so that one create the cause or problem to section which one is much useful our operation area. And our research question when we're starting to that research, the classification, how the classification to AI tools and distribution, how it's changed to time period, trends, what is the new trends or software architecture, similarities between the AI tools and decision supports, how we can use. And I simulate two different kind of disaster. One of Alzheimer disaster. If we check first step literature or create a hypothesis, that if you are researched it steps, you can use BERT, BioBERT, BioGPT, BioBART. That kind of AI you can use. But if your focus area dynamics or mutation discovery, you can use Dragon and GATS. You can predict DNA mutation. In the first of all, where can be potential mutation, or how, and how is the change. Also, you can predict that question. Uh, Another one, transcription of counts. For example, if you are working to wet lab, if you want looking for the level of protein on your samples, you can use Solomon, Callisto, RISAM, Desquient, HGR. Not only for protein, also DNA level you can measure by the using that kind of tools. Different kind of steps, different kind of tools. Also different kind of disasters, different kind of tools. For example, a cancers disaster, and the first step maybe imagination is very important because different kind of image there are events. For example, tomography, Röntgens, or fMR, MR. So all kind of some of video, some of only image. So different kind of input data, different kind of operation area, different kind of tools. Digital pathology, tumor genomes, drug discovery. It's very important. Also AlphaFold. One slide back, you can see same kind of tools. AlphaFold, also Solomon, I mentor. So maybe disaster is the change, but same tools. Sometimes disaster is the same, but different kind of steps, different kind of tool I share with you. Methodology, and the first of all, we determine our research area. We check the 153 different kind of AI tools. After then, we determine 25 different kind of parameters for elimination or determine the clusters. We use Euclid's distance and methodology word hierarchical clustering. Here is 14 different kind of classification for determining 153 different kind of tools. For example, SQAM, structured biology. There is here approximately 8 different kind of AI tools. Here, protein language models. Here is visualization, molecular dynamics, system biology, evolution, microbes, transcription, RNA sequences, single cells, protein structure, prediction to design, especially AlphaFold here. You can see that's very popular, and programmatics, metabolics, mass, genome, artificial intelligence, clinical NLP, image process here, BioBERT, PubMed, BERT, that kind of tools. molecular docking, drug discovery, and others AI, not specific operational and optimization tools. You can access the details of all app or tools and the main documents. Here's trends that's starting the 210 and finishing the 226. In the first 5 years, actually, it's not so much differences, but suddenly 100, 215 and suddenly 218, two different dates. It's very critical improving to bioinformation AI tools. First reason, here is the statistical model cover to machine learning models. That one is the red one is the most effective. This return changing. That's optimization tools. Other critical position, 118. That one is the one architecture of software. Name is transformers layering generated by the goggles. Now we use that tools methods for protein language models, and other one CNN, that one using generally for image processing. So two technology to new software effective AI tools. After then, new area affect generative AI. I share some examples there. That one we can create some simulation, driving mid-journey methods using generally. For example, AlphaFold, we can imagination 3D structure of proteins. Here again, I sharing the two different critical dates. That's very important. CNN, that's graph neural network. That one, not only one type of data using different kind of data can be integration. Your models, for example, if you create the model drug and side effect problems, that one heterogeneous graph, so you can modeling the disaster modeling. So now it's very popular, should be increased popularity is the next years and the future actually. Here are 25 different kind of criteria. First of all, we ask some question our research space. First of all, what is your organization focus? Generally, human-centered 53, only human data using by the AI tools, ratio 53. General taxonomy is mean, for example, proteins, not discrimination to plant or animals or human or microbes. Just interesting to protein structure, so that's mean general taxonomy. Only plants, no any tools interesting. That one inside the general taxonomy. Model organization, that one generally evaluation models. For example, maybe just interesting mammals or just interesting flies. So not only human or animals, only flies, only mammals. So both of possible, but not all. 5% microbes. Here is the 5 different kind of criteria. Here is the 9 different of criteria for what? For train or input data type. How is creating that AI tools? 18% only DNA and RNA data use. 11 is single cell transcription data use. 10% mass data use. 16% protein structure 3D data use. Chemical structure 8% use. 7% clinical test AHR data use. Metagenomes is 5%. Medical image 14% data using to creating health AI tools. Another question, what kind of AI models architecture using the bioinformatic AI tools? NLP models 19, 18% CNN image process. It's very similar because it's the both is starting to 218 years since. So similar perspective. One of the protein language, other one image process. But most of ratio creating by classical machine learning statistical method, because it is starting 210 until to 218 only we use classical machine learning and statistical model. So maybe 153 different kind of AI tools, approximately 75 tools only use a classical machine learning or statistics model. New area, graph neural network 6%. Reinforcement learning, we use computer science to games marketing, game marketing, but this architecture not using the bioinformatics AI tools. Also, PNME3, that is mean signal transmission models for brain synapses logistics. That one 6%. And the last question, what is your prominent operation area? 46% analyzes tools. What is the mean? Maybe you share your sample data, and then tools measure to protein level or DNA level. Other one prediction, 38%. That one predict the docking or binding. Other one is generative AI. Looks like AlphaFold style imagination, the 3D structure over the space. And last one, simulation, 10%. It's very critical process because of, they are interesting environment condition for binding. For example, if you increase the temperature, how it affect the binding? If you decrease the press, how is change the binding? Or ions condition, salt condition, water hydrography condition affect the binding. So some level increase, some level is the decrease. You can simulate the binding condition by using optimization AI tools. That one, hierarchical clustering graph, maybe font is so small, maybe we can read, but you can access the detail, the main text. I want to show you here how you can use this one. If you look to bottom of the graph, you will see a similar kind of AI tools, because the distance is this 0 according to 25 criteria. That is mean both of you can use your operation area, so you can cross check or validation both of result and your result. So that position you can validate with use that graph. A conclusion, systematically classification 153 bioinformation AI tools, we using 25 different criteria. Develop a comprehensive classification framework for AI tools in healthcare and bioinformatics, integration technical characteristic application domain and AI architecture into the single taxonomy, help the researcher identify alternative tools with similar technical capacity. Just one slide before I explained it, how supported efficiency based on AI tool selection, different research objective. Also, beginning of my presentation, I share Alzheimer and cancer examples. Improve the interoperability of the AI ecosystem, especially the wet lab researcher. It's very important for wet lab researcher. Enable cross-validation by identify technical similarity AI tools. That is bottom of the graph. Facilitated AI follow-up planning across the different stages of biomedical research. Also, I share big of the cancer, Alzheimer disasters, different kind of level, different kind of AI tools promote the more efficiency information adaptation AI in bioinformatics. I hope this presentation helped to this area. And thank you very much for your listening.